McMillan was on the gym. Absolutely. The one who was. Yeah, they have a ravine. You go like under the bridge. My old school, Northland College. It's in Ashland, so like. I'm gonna say she's right, like walking out to the woods. What? Yep, Northland College.
All right. Morning.
Good morning.
Okay. All right, so we have a few things to talk about this morning, and then we need to finish up just the last tiny bit of chapter 5 and move into chapter 6. So I'm going to start off by sort of our standard review of things that are coming up. Chapter 5 homework due Friday. How are those going?
Yeah, it's a long
They're okay.
I turned it in last time.
Okay.
It's hard,
It's okay. Okay. It is kind of tough. We've entered into sort of a different type of material, and applying it can be a little bit challenging. I will have time on Friday in class to come around and see how everybody's doing on that. If you have other questions or want me to clarify other points, please come by my office and or find somebody who has already come by my office. Okay, so use each other as resources, use me as a resource. Together we can succeed. Okay. So, I think the next chapter's homework, chapter 6, is a little bit easier in that it's more definitional and less math. Okay, so on this next exam we have one chapter that's very math heavy, and then we have one chapter that's more recognize and know heavy. Okay, so kind of a nice balance. So chapter 6 worksheet is currently due on Wednesday of next week, which is the day of the exam. I don't see a point of extending it because then I think that people will wait to work on it, and I don't want people working on it over fall break. That just makes silly to me. So I don't want you thinking about genetics while you should be taking a break. We all need breaks. So, okay, so between now and next Wednesday, we do have a fairly heavy genetics load though. Genetic load, that's kind of like a joke, but we haven't gotten to genetic loads yet. So. It's got mutational accumulations. All right, anyways. Now I'm distracted. Thinking about genetic loads. All right. No, it does not sound right. Kind of related to what it actually means. Okay. So, chapter 5 homework, Friday. I teach a lot of random things. So. I am teaching a gonads class. I do teach anatomy and physiology. None of this stuff is weird or embarrassing to me anymore. Okay, chapter 5 homework Friday, chapter 6 homework Wednesday, exam 2 Wednesday. Exam 2 is 2 chapters long. I have it with me, so I can give some guidance on it. I do have the exam. Yeah, right. I'll just change the questions. Okay, so 12 multiple choice questions, less than half of the last number of multiple choice questions. 4 short-ish answer questions. I've already told you what one of those is. Yes? Okay. That one's worth 20% of the overall exam grade. So make sure that you know how to do questions 8, 9, and 10 on the homework. Those are your guiding questions for this question on the exam. You will also need to know how to take information from a test cross and determine if it is linked or unlinked. So I'll give you test cross data. And you'll look at the test cross data and tell me whether it is significantly different from what we would expect under independent assortment to the point where we may think these genes are linked. 15% of the exam. Okay. The other 2 short answer questions are information that we'll cover in the next chapter, so chapter 6. It'll be the 2 major concepts from chapter 6. So I don't want to tell you now because it's not going to make any sense, so we'll come back to that at the end. Okay? Questions on that? Okay, so you should be able to focus your studying. There are still multiple choice questions, so you'll still need to know like the basics from chapter 5. It's not just can you do a linkage problem and can you do a chi-square problem. It's you know the basics and can you apply them. Okay? All right. It should be much shorter than the last exam. If you know how to go into a question, so you see a question, you can automatically get it started. Those questions shouldn't take you more than 5 minutes apiece. What else did I want to tell you? Chapter 5 homework we talked about, exam we talked about. On MO2, I have posted under week 6 a questionnaire. It is called a mid-course evaluation. Okay. I ask that if you are willing, you fill it out for me. I can show you what the questions are. It just asks, how long you're spending preparing for this course. If you agree with a series of questions about an instructor, me as your instructor, if you agree or disagree about a series of questions about the course and course content, and then what resources or materials you find useful, and which ones you actually like. Okay. And then there's places for comments. What do I do well? What can I work on? This is not a requirement of anyone, but it helps me alter things for the second half of the semester so that I can gear things more towards what you all need from me. So the first half of the semester, we're kind of learning what or how we interact. The second half of the semester, I can adjust certain things to make more sense for you all. So if you're willing to fill this out, I appreciate it. It's entirely anonymous. I can't see who's done it. I can't see who says what. Okay. So there should be no concern. I think it says that it's anonymous on your end. I don't know if it says that on mine. I'm not even eligible to take it because, actually, is somebody on MO2 now? Can somebody with a computer try and log in and click on this and see if it lets you take it?
It's in your week 6.
It's under week 6.
It's under week 7.
Yeah.
Oh, really?
Yeah.
Mine says
Mine's on 6.
Yeah, mine's on 6.
Well, why is mine on
Yeah, you can
7?
go.
Oh, now it's moved.
Yeah, you can get into it. You can log into it and you can access it. Okay. So if I get, how many students are in here? 25 total. So if 22 people are willing to fill this out, and again, it's entirely anonymous, so I don't know who those 22 are, I will give extra points on the next exam because this information is really helpful for me. So it shouldn't take more than like 5 minutes, and really it helps me make things better for all of us in the second half of the course. So I appreciate if you're willing to take the time. I like to incentivize it because we all say we're willing to do it and then we forget. I do that all the time.
Sounds like every course evaluation for quality that he asks.
Exactly. So if I incentivize it and I ask you then to pester each other, because I don't know who hasn't done it, but you guys can ask each other who's done it and who hasn't, then I get a little bit better response rate. Okay? Okay. All right. Any other questions about what's coming up? Everybody good? All right, so we had just a couple of slides at the end of this chapter that I wanted to decide if I wanted to cover or not. All right, so I'll just quickly, very, very quickly talk about this concept of mitotic recombination. So as we've talked about linkage and crossing over and recombination, we've always talked about it in terms of gamete production and genetic variation in the next generation, right? We have not talked about recombination in like somatic cells or body cells. It's pretty rare that this actually happens, and that's because if we think back to mitosis as compared to meiosis, in mitosis we don't get tetrads lining up, right? And so when those tetrads line up is when we get that crossing over between those homologous chromosomes. In mitosis, we don't get tetrads lining up. So it would be pretty rare for recombination to actually occur. It does happen very, very occasionally. Usually it's very early in embryonic development, so it would be unlikely that it would happen in adults. Usually if something like this happens in adults and we get, so in order for crossing over to happen, we have to get a double-stranded DNA break, which means that we have to cut both strands of DNA on a chromosome and then realign them and put them back together. Usually if we have double-stranded breaks in our somatic cells, so again in our body cells, that cell just goes through apoptosis. Double-stranded break is usually considered a fatal error, and so that cell would sort of die off. In early embryonic development, we're more likely to get these types of breaks and sort of recover from them. In this case, this is just an example where we have two different genes on the X chromosome, right? So you imagine an individual has this combination on one X chromosome and this combination on another X chromosome. If we get recombination, we actually change the phenotype of the cells. Okay, so when they're here, we have dominant alleles in both of these chromosomes. I'm sorry, in this chromosome. So in this cell, we have a dominant wild type Y and a dominant wild type SN. Right? If crossing over occurs, so here's mitotic crossing over, we end up with daughter cells that get the wild type of one allele and the recessive of the other allele. So this daughter cell and this daughter cell do not look like the parent cell that it came from, because we had a crossing over event that changed the allelic combination within that daughter cell, and so we end up with two phenotypically different cells. And so this can appear kind of similar to exon activation patterns, where we have sort of patchy distributions of different genes being expressed. Often, mitotic recombination is going to result in what we call a twin spot. So if you take a genetic sample from sort of this little tiny yellow affected area, it would actually have a different allelic combination than the rest of the body, right? So it happens very early in development. So this cell then proliferates and gives rise to a whole bunch of cells that create an area that is phenotypically different than the rest of this, in this case, form. Okay. So that's about all I wanted to cover with that, just sort of a quick rundown. Any last questions on crossing over, recombination, yada yada? So I actually want to give a quick clarifying point before I move on. In questions 8, 9, and 10 on your homework, I did not ask you to give me like parental genotypes of those, or F1 genotypes, or test cross genotypes. Is everybody comfortable with determining what those are? So if I were to give you, I'm just going to pull up the homework real quick. I just want to make this very obvious. It can be confusing when I say parental genotypes, and I think this came up in lab yesterday. The difference between a parental genotype and the parent of a particular cross are often different. So in this case, I've given you an example where we have 3 genes. Okay, so we have a parental generation. What's the parental generation in this case? And also there's an error on here. This should be a capital S. Correct that. Nope. Nope, that's wrong. This one should be an S, not A. I knew there would be errors. It's a mutation. That one has a mutation that occurred. Okay. Okay, so what are the parental genotypes? If I say, give me the parental genotypes for this problem, what would they be?
All the biggest numbers.
The biggest numbers, yeah. So the parental generation is actually this. This is one parent. I'm sorry, that's a capital. And the other parent would be this. So for these, we should try and use chromosomal notation as opposed to sort of the standard this version. So that's how we would have represented it in the past. This essentially indicates that these are unlinked. So since we know that we're dealing with a linkage problem, we should put it in linkage, in chromosomal notation. Is everybody okay with that? Okay, so when I ask what the parents of these offspring are, what were the parents for this cohort's, what were the parents of this cohort? So what were the genotypes of the parents from this cross? Okay, so an F1 who would be heterozygous. Yes, and the other one is homozygous recessive. So this is the result of a test cross, the progeny of a test cross. So we automatically know that one of our parents has to be this. And the other one is a member of the F1 generation, which gets this from one parent and this from one parent, so they become that heterozygous. Does that make sense to everyone? Okay. So I know that sometimes we think, oh, the parental generation is this, because these are the parents of this set of progeny. But when we say parental genotypes or parental phenotypes, we're talking about these guys, the parental generation. Okay? So just make sure when you're working on these that you can distinguish between parental, F1, and test cross. This information should come from a test cross, which is an F1 and a recessive. Okay, just want to make sure that that was clear before we move on. All right. So now we are going to move into chapter 6, which is a chapter about cozy sweaters. No, about variation in chromosome. We're talking about structure and number. So these are going to be really large scale changes in chromosomes. We have kind of micro changes, which would be changes in an allele or changes in one nucleotide within a chromosome. We also have really big chromosomal changes, which are going to change the genes that are present, the structure of the chromosome, or actually the number of the chromosome entirely. So, I like to talk about bananas here. So a lot of our fruit, what is weird about a lot of the fruit that we buy in the store? Like, what is weird about a banana or a watermelon or...
Doesn't come from here?
They do not come from here.
Are they really genetically different than these like originally were?
Yeah, so they're very genetically different from sort of their ancestors. That genetic difference is often associated with the number of sets of chromosomes that they have. How many sets of chromosomes do bananas have? 7. Although grapes are octoploid, so they have 8 sets of chromosomes. Onions, I think, are octoploid, the ones that you buy in the store. Plants are weird. Yeah. So bananas, most of the bananas that we buy in the store are triploid, which means they have 3 copies of every chromosome. How does that work in meiosis? How do you evenly divide 3 sets of chromosomes?
Is it impossible to divide pretty evenly?
Yeah, you're never going to get gametes that make sense chromosomally.
Okay.
So what is not found inside of a banana? seeds, right? So we do things like breed triploid bananas that are seedless, because who wants to eat a banana seed? Which is good for us, but how do we make new bananas?
Oh. The seeds are low.
The answer is not for genetic loads. All right, I'm gonna play a video about bananas now. Actual bananas. And sweaters. I find this fascinating.
If you like bananas, you probably eat a lot of clones, because all bananas are clones, or at least Cavendish bananas are, and they're the cultivar or variety of banana that most people eat. They're genetically identical to one another. And they are also all in danger of being wiped out by a fungus. Cultivated bananas reproduce asexually through a process known as parthenogenesis, which literally means virgin birth. Most plants have male and female flower parts and seeds that are pollinated with the help of things like the wind or bees. Pollination is the plant version of getting it on. And there are ways to create new banana cultivars if you pollinate them by hand, but it often fails, and even when it does work, it's hard to know if your new banana is going to look good, taste good, and be easy to grow. So most banana plants live a chaste life. If you're a banana farmer, you get more banana plants by cutting off a piece of the stem of an existing plant and then sticking it in the ground. That plant then matures, blooms with potentially more than 300 bananas, and then dies. Because they're all the same, they're super vulnerable to disease. There's no chance that one banana plant will happen to have a gene that makes it resistant. So if a parasite or fungus can kill one banana, it could potentially kill every banana of that type. That's something that's happened before. Back in 2013, we here at SciShow talked about how the Gros Michel banana, which was bigger, sweeter, and hardier than the Cavendish, went virtually extinct in the 1950s. The culprit was a highly infectious fungus we called Panama disease. In the space of about 10 years, Panama disease reduced the Gros Michel from the world's most popular fruit to just another weird thing your grandparents talk about but nobody else remembers. And the banana industry would have died with the Gros Michel if it weren't for the last-minute switch to the Cavendish. Well, it wasn't as big and it didn't taste the same as the Gros Michel. The Cavendish was immune to Panama disease, at least the type of Panama disease that was around at that time. In 2013, we also told you that a new strain of Panama disease had emerged that could infect the Cavendish, but that it would probably be fine. Infected fields were being quarantined, scientists were working on developing a new kind of Cavendish that could resist the new disease, and hey, we have the lessons of history. We weren't going to let what happened to the Gros Michel just happen again, right? So here's an update on that story. According to a study published in November in the journal of Pathogens, this new strain of Panama disease, known as Tropical Race 4, or just TR4, is spreading. It originated in northern Australia, and the last time we talked about this, it was also in Southeast Asia and southern China. Since then, the researchers note, TR4 has reached the Middle East through Jordan and Lebanon, as well as Africa through Mozambique. TR4 in Africa is especially concerning because bananas in many parts of Africa, including Mozambique, are not just a tasty treat, they're a staple crop and a major component of national food security. Even worse, it seems likely that TR4 could also infect other varieties of African banana, many of which are grown as subsistence crops by rural families. What makes TR4 so dangerous? It's caused by a fungus that spreads through soil. If you didn't think dirt could carry disease, it can, and when it does, it's bad. TR4 spreads through the...
Sorry, I had a different video that was about, let's try. That wasn't the one that I thought. Oh, reggae rock, seedless banana tree. That was not good, that's more evolution and...
Here at SciShow HQ, we have a little food area for the employees. Sometimes there's donuts, sometimes there's nuts, sometimes some dried mango. But the one thing that never sticks around and is gone as fast as we can buy it is the wonderful, beautiful, noble banana. Unfortunately for us, they may not be around forever. First, the good. Bananas are healthy, packed with nutrition and energy. They fit in your hand and give nice little cues when they're perfectly ripe and are easy to peel and eat. Shocking statistic, the banana is Walmart's #1 selling item, not the potato chip, not Coca-Cola, not 50 Shades of Grey, bananas. They appear to be so perfect for human consumption that Kirk Cameron attempted to use them to prove the existence of God. Of course, this banana was not created by God, or really even nature. Bananas, at least the ones that you see at the store, were created by people. Don't get me wrong, there are wild banana plants, lots of them. They're native to South and Southeast Asia, and there are dozens of species and thousands of varieties. They're just not the ones we eat. Some of those species, as you might suspect, have seeds, because that's what fruits are. They're fleshy bodies containing seeds. So you might wonder, why have you never eaten a banana seed? Well, you have, kinda. In cultivated bananas, the seeds have pretty much stopped existing. If you look closely, you can see tiny black specks. Those are all that's left, and they're not fertile seeds. If you plant them, nothing grows. Today's bananas are sterile mutants. I'm not trying to be mean, that's just the truth. Unless you were alive in the 1960s, hats off to all those older SciShow viewers out there, every banana you have ever eaten was pretty much genetically identical. This is a Cavendish, the virtually seedless variety that we all eat today, but it wasn't always our banana of choice. Until the 1960s, everyone was eating the same banana, it was just a different banana, the Gros Michel. A bigger, sweeter fruit with thicker skin. You might notice that banana-flavored things don't really taste like bananas. Well, they do. They taste like the Gros Michel. The genetic monotony of the Gros Michel crop was its undoing. A fungicide-resistant pathogen called Panama disease began infecting the Gros Michel crop. By the time growers understood how vulnerable their crops were, the Gros Michel variety was all but extinct. The entire banana industry had to be retooled for the Cavendish. Since they're seedless, the only way to reproduce them is to transplant part of the plant stem. And for the last 50 years, we've been good with the Cavendish, because it's more resistant to the Panama disease. However, somewhat terrifyingly, a strain of Panama disease that affects the Cavendish strain that we all eat has been identified. A global monoculture of genetically identical individuals is a beautiful sight to a pathogen. The fungus only has to figure out how to infect and destroy a single individual, and suddenly there's no diversity to stop it or even slow it down. That's led to a lot of scientists worrying about or even predicting the outright demise of the Cavendish. This wonderful, most popular of fruits might completely cease existence. The good news is we now have a much better understanding of genetics, epidemics, fungi, and pathology. Scientists and growers have already taken steps to protect the Cavendish. Some growers are creating genetically different bananas that might replace the Cavendish crop if it fails, while scientists are attempting to genetically engineer Cavendish plants with immunity to Panama disease. Plus, we learned a lot from the Gros Michel debacle. Infected fields are quickly being destroyed, and new crops are grown from pathogen-free, lab-grown plant stock. So thanks to the people who work tirelessly to grow and harvest bananas and bring them to us so that we can offer them inexpensively to our employees. And thanks to the growers and scientists working tirelessly to make sure that they don't go the way of the Gros Michel. Thanks for watching this episode of SciShow. If you have any questions or comments or suggestions.
All right, so one of the applications of sort of genetic modification is in our agriculture. We do it all the time. And then we produce things like bananas that don't have seeds, which most of us enjoy. I don't want to eat, that banana with seeds is kind of scary.
I think of a banana at first.
Yeah, it doesn't look like a banana. I mean, the same is true when you look at sort of a lot of wild cultivars, like, you know, corn compared to its wild ancestor looks nothing like corn that we have today. But next time you eat, are runs still around? Runs? Yeah, runs. Next time you eat runs, that banana flavor is what bananas used to taste like. So maybe if our current bananas go extinct, they'll change it to memorialize our current banana. Okay, so we are going to talk about sort of two pieces here. First, we're going to talk about chromosome structure, and then we're going to go back and talk about number, and we're going to look at polyploidy and different combinations of chromosomal sets within an organism, which is what happens in plants most often. All right, so here's a list of objectives. That's a lot of things. I don't feel like reading them. So or as we go into this, we are going to start talking more in a couple of weeks about genetic variation and how we sort of quantify or assess genetic variation. There is genetic variation within a species, which would be intra-species genetic variation. So when we're talking about humans, I think we all know at this point that we can tell individual humans' DNA from each other based on variation. Right? My DNA doesn't look anything like, well, it looks something like, but anybody else's. right? There are ways that we can differentiate each person's unique genetic composition. There's also interspecies variation. Again, we can sort of tell these at the sequence level. So if we just take a gene and its A's and C's and T's and G's and compare them, they will likely be different between two species, although still similar. We can also look at chromosomes, right? So we've talked a lot about how humans have 46 chromosomes or 23 pairs of chromosomes. There are many other species that have more chromosomes or less chromosomes. Okay, so we have rhino species have like 82 chromosomes. Fruit flies have 4 or 4. Okay, so there's a lot of variation in how many chromosomes exist. That is going to be interspecies variation, so between species. Within a species, most individuals have the same number of chromosomes, have the same sort of DNA length, same genes. All right, so mostly what we're looking at here is going to be chromosomal abnormalities, which are going to be changes in the structure of a chromosome and changes in the number of chromosomes. So if we're looking at this karyotype up here, what is our chromosomal abnormality? Enter.
Number 10 has 3.
Yes, number 10 has 3 copies. Okay, everything looks okay until we get to number 10 where there are 3. This is going to be fatal. We cannot survive with 3 copies of chromosome 10. 12 also has 3, yes.
It's not fatal.
It would also be fatal. Yes. There are very few examples of non-lethal triploid chromosomes. 13, 18, 21. 13 and 18 are usually fatal by a year of age. 21, lifespan is fairly normal.
Would number 2 be a structural?
Say that again?
Would the number 2 chromosome be a structural? In a ball, or is that just?
Possibly.
It's like rolled up.
Sometimes when they're doing karyotypes, chromosomes just like fold in on themselves. So there could be something there. It would actually be hard to tell without sort of uncoiling it and looking at what's going on. Okay, we identify chromosomes based on a couple of things. So there are 3 criteria which help us sort and align chromosomal pairs when we're looking at a karyotype. One is location of the centromere. And so these 4 examples down here are how we assess chromosome or centromere location along our chromosome. So if the centromere is basically in the center of the chromosome, we call that metacentric. And when we're determining if it's in the middle, we are going to look at if the P and Q arms of the chromosome are the same length. So P actually stands for petite, so usually the short arm is going to be labeled P. Obviously these two look similar in length. There is one that is probably technically shorter than the other just based on the number of nucleotides, so that would be the P arm. So P, Q is going to be the long arm. If we look at sub-metacentric, you can see the centromere has shifted up a little bit. Now we have a pretty distinct petite arm or P arm and a much longer Q arm. Acrocentric is an even higher shift in that centromere. And then telocentric is essentially a centromere that is at the ends of a chromosome. There would be very small segments of DNA sticking out of this side. So there is a petite arm, but it's mostly telomeric DNA. I would suggest highly that you are able to recognize metacentric and acrocentric at the least. Telocentric is pretty easy. Metacentric can be a little bit hard to differentiate from metacentric or acrocentric. So I will talk about metacentric, acrocentric, and telocentric most often. Size is another one which we've talked about, and banding patterns we've also talked about, right? So size is simply the number of the number of nucleotides in a chromosome. So when we look along, again, a human karyotype, you'll notice that our longest chromosome is 1. That's how we order chromosomes. We start with the longest ones, and we work our way down to the smallest ones. And then banding pattern is determined by a series of different dyes. So if you look, again, along this karyotype, you'll notice that along these chromosomes, there are dark regions and light regions. If you look at 10, you'll notice that the dark regions should occur at the same sort of locations and the light regions should occur along the same locations. So if you put 10 next to 9, this dark region would line up with that light region. That's how you can tell that those are not the same chromosome. So this is the banding pattern that people who are actually looking at these chromosomes sort of look for. You can tell based on the banding patterns what chromosome they're actually looking at. Okay, so we have a number of different types of chromosomal mutations, everything from insertions and deletions to inversions, translocations, and changes in total chromosome number. So we can kind of classify these. Here is an example where we have a rearrangement of the chromosome. So if we just look at this top blue chromosome, this would be normal. Okay. Here, in this first example, the green chromosome and the peachy chromosome look correct. But what we now see is that in this blue chromosome, one of the copies has extra genes. Right? So we have A and B, CDE, CDE. So this is an example where we've had a duplication event. We still have the same number of chromosomes in the cell. What we've changed is the number of genes that are present. So instead of having 2 copies of A, 2 copies of B, 2 copies of C, D, and E, we have 2, 2, 3, 3, and 3. Okay, so we've added additional genetic information. Here is an example where we have a trisomy. A trisomy would be where we have an extra copy of one entire chromosome. This is polyploidy, where we have an extra copy of every chromosome. As we go through each of these sort of processes or these mutations that result in changes to chromosomes, I want you to make sure that you can note whether the total amount of DNA is changed or whether it stays consistent. So whether or not we change the total amount of DNA in a cell is going to help us figure out if the impact of that chromosomal mutation is severe or not even noticeable. Okay, so we're going to go through these. It should be pretty obvious, but just make sure that that's sort of a piece of information you're keeping track of as we go. Okay, so looking at chromosomal structure, we're going to talk about 4 processes that change individual chromosomes, or sets of chromosomes. The first one is duplication, second is deletion, and then we'll talk about inversion and translocation. So I'm going to show them up here now and talk about them generally, and then we'll go through each one individually. So duplication is what we just looked at on the first slide, where we have a chunk of DNA within a chromosome that is duplicated. Right? So again, we have A, B, C, D, E, F, and then we have E, F, and G. So this section, E and F, is duplicated, and now we have 2 copies of E and 2 copies of F within this chromosome. On the homologous chromosome, we'll still have just 1 copy of E and F. So in a cell in its entirety, we will have 3 copies of E and 3 copies of F. So for a duplication event, what is happening to the total amount of DNA in the cell? Increasing, decreasing, or staying the same?
Increasing?
How many people feel like it's increasing? How many people feel like it's decreasing? How many people feel like it's staying the same? Okay, it is increasing. Right, so we can imagine that these are the homologous chromosomes in the cell now. we have extra base pairs. We have a whole extra section of DNA that's literally been copied and pasted. Right? We copied it from here, and we accidentally pasted it in there again. So when we have a duplication, we increase the total amount of DNA in the cell. Specifically, we increase the number of genes within one chromosome. All right, let's try this with the next one. So deletions are going to be removal of a section of DNA. They can be removed from sort of within a chromosome. So in this case, again, we're removing E and F, so we have A, B, C, D, and G. You could also remove sort of the end piece of a chromosome. So sort of two different options for how deletions work. What's going to happen to the total amount of DNA in this cell? Decrease, right? We're cutting out genes. So now inside of a cell, we are only going to have one copy of genes E and F. For an inversion, we take a section of DNA and we flip it 180 degrees and reinsert it. So this is our original chromosome, A, B, C, D, E, F, and G. The resulting chromosome from an inversion, A, B, C, F, E, D, G. So what happens to the total amount of genetic material here? It stays the same. We still have two copies of A, B, C, D, E, F, and G. Okay, so we have all of the same information, we've just reversed its order. And then a translocation can get a little bit tricky. There are different types of translocations. And the one that's shown here is what we call a reciprocal translocation. So we have two non-homologous chromosomes. So this would be chromosome 1 and this would be chromosome 2. Usually, we should not exchange information between non-homologous chromosomes ever. It doesn't generally work very well for us. But it does happen on occasion, so we break off the ends of these chromosomes and flip them. So what was A, B, C, D, E, F, G becomes A, B, C, D, Q, R, S. And then we have M, N, O, P, E, F, G. Okay. So in this case, what's going on with the total amount of genetic information? It stays the same. Right? Again, we're not losing any genes, we're not losing whole chromosomes, we're not removing genes, we just changed where those genes are occurring. Okay. You have just answered part of another exam question. Good job. Okay, and it's also on the slide. Okay, so these are definitions for each of the things that we just talked about. I'm not going to read these to you because I'm pretty sure you can read them. The only one that I'll point out is for translocations because we do have different types of translocations. The one that was shown on the previous slide is that reciprocal translocation piece down here at the bottom, where we have two non-homologous chromosomes, both breaking and trading pieces. This one here, simple translocations, would be if a chromosome broke off part of itself, or a chromosome broke, and that piece merged with another chromosome with no exchange of material. Again, the amount of genetic material in a cell is the same. We've just changed one chromosome becomes shorter and one chromosome becomes longer. Does that make sense? All right. Now people are copying these down, so. Thank you. Okay. Happened a long time ago, my dogs ate, or tried to eat. I came home and there was, wait, where? came home and my cactus was on the floor and I was like, oh, that's sad, one of the cats knocked it over, and then I looked at it and there was like a dog-sized mouthpiece taken out. And then so like over here was the plant, and then over here was the chunk that they had like bit off and then left. Why would you try and eat a cactus? They're not very bright. It's weird, they're like smart when they want to be, but also not very bright. Alright, so I think the last couple words are good. Okay. Okay, so for deletions, you might want to get comfortable sort of drawing out what these things look like. So again, I mentioned we have two different types of deletions. When I'm asking you to draw a chromosome, all you have to do is draw a line and then put like, yeah, that's all I'm asking for if I'm asking you to draw a chromosome. It doesn't have to be art. If we had a terminal deletion, that would end up resulting in a chromosome that looks like this. So terminal is off the end. We would lose the D. Interstitial is what's being shown on this slide. Interstitial would be where sort of the end pieces of the chromosome remain intact, but we lose material from the middle of the chromosome. But either way, if we have a deletion occurring, we are losing some genetic material from some part of this chromosome. The consequences of a deletion in terms of phenotype, so what result is this going to have on an individual, can vary wildly. Part of that is going to depend on the size of the deletion. Okay, so the deletions I drew on the board were one gene each. That one gene may have a necessary function. So if you deleted a gene that resulted in production of actin, if you don't produce actin, none of your muscles can move. Okay, that's going to be a fatal deletion, a deleterious, unlethal deletion. If I deleted a gene that coded for knuckle hair, probably not going to kill you. Okay? So it all depends on what is being deleted and how much of it. Right? So if you delete half a chromosome, chances are that's not good. If you delete one little section that has a gene on it, potentially okay. We end up with, when we delete a gene, we get what's called gene product imbalance, right? So everybody in this room presumably has 2 copies of every chromosome, which means that in all of our cells we have 2 copies of every gene that is necessary to survive. If you delete a gene, you likely still have a copy of that gene on the other chromosome. So deletion doesn't mean that we delete that gene from the entire genome, it means that we delete it from one chromosome. So in a cell, after a deletion takes place, you would have this combination of homologous chromosomes. Right? So we still have that gene present, we just only have one copy of it. 2 of the major issues that result from only having one copy of a chromosome are pseudodominance and haploinsufficiency. So pseudodominance would be when a recessive allele is no longer masked by a dominant allele. So let's, for example, say that allelically this copy was recessive and it was a loss of function gene or something. The one that was deleted was a dominant allele. In this case, we now have pseudodominance. That recessive allele looks like it's the dominant one because it's the one being expressed, because we've lost the other copy. We've lost our dominant copy. So that's one potential outcome. Another one is what we call haploinsufficiency, which is that last word there on the slide. This would be when that one copy of an allele does not produce enough protein to result in a wild type phenotype. So again, we'd still have, in this case, we'll just make them, this will be a dominant allele again. Even having this copy of the dominant allele, you only get maybe 50% of protein production. That may not be enough protein to actually give you a wild type phenotype. So all of these things come into play when we're trying to determine whether or not a deletion is really an issue or not. Again, some genes, not a huge deal, other genes, big deal. Okay, we will pick back up with the rest of our chromosomal alterations on Friday. I'll probably lecture we'll talk for about half of the class, and then I'll answer questions on the worksheets, either of them. Saturday? I'm trying to decide in my brain, but I realize I'm probably not going to be awake at midnight on Friday. So Saturday, I will post the answer key to worksheet 5. If I forget and you're looking for it, email me, please. I will probably forget. I'm just going to throw that out there. Email me, and I'll post it.
Saturday. Got it.
I'll ask them what's
Emailed that late
last night?
last night, so.
I'm probably not gonna print it. Bye.
Somebody left the charger.
No, not mine.
I'll take it. Just kidding.
I'm totally kidding.
Someone will realize.
Somebody will need it too.
Should I tell a story?
Oh, what?
There's a one day it's like this story from it.
Oh, yeah, sorry. That's okay. Up here,